Have you ever stopped to wonder what a thought physically is, or how a memory gets stored? Today, we're diving deep into the very spark of consciousness. We're going to peel back the layers of the brain to understand its fundamental language, which is all about electrical signals and the incredible cells that create them. Let's get into it. It's no exaggeration to say the brain is one of the most complex things in the entire known universe. It's the engine that builds our reality. But here's the amazing part. All of that complexity, every single thought, memory, and feeling, it all boils down to a set of really elegant biological and electrical rules. So to really understand the whole picture, we have to start with the smallest parts. So here's our game plan. We'll kick things off with a big picture, looking at the brain's evolution. Then we'll zoom way in to meet its cellular workforce. From there, we'll uncover how neurons get charged up, how they fire off signals, and the seriously clever tricks they use for speed. And then we'll wrap it all up with the core principles you need to know. To really appreciate the brain, we first have to get a handle on its sheer scale. The numbers behind this organ are just staggering, and they tell this incredible story about evolution and energy. Okay, let's start with just pure mass. The average adult human brain weighs somewhere between 1,300 and 1,400 grams. That's about 3 pounds. So yeah, it's a pretty significant piece of biological hardware, for sure. But as you're about to see, its weight is only a tiny part of the story. Because here's what's really wild. That 3-pound organ only makes up about 2% of our total body weight. It's an unbelievably compact package. But don't let that fool you. This thing is incredibly demanding. Tucked away inside that small space are an estimated 86 billion neurons. Just think about that number for a second. That's more than 10 times the number of people on the entire planet. And each one is a tiny little processor, all networked together. And this is the punchline to that 2% figure. Despite being so small, the brain is an absolute energy hog. It chews through a massive 20% of your body's oxygen and calories when you're just sitting there at rest. That tells you something really important. It's always on, constantly managing a ridiculous amount of background processes, even when you're not actively thinking. This journey through evolutionary time gets really clear when you start comparing brain sizes across different species. You can see this dramatic trend, right? As animals get more complex, from a rat to a cat to a primate, there's this huge increase in brain size relative to their body size. Let's put a hard number on that. If you look at our closest living relatives, chimpanzees, their brain volume is around 350 cubic centimeters. The modern human brain, it's sitting at around 1,350. That's almost 4 times larger. In evolutionary terms, that is a colossal leap. But it's not just about getting bigger. It's about where that growth happened. The expansion is almost all concentrated in one place, the cerebral cortex. That's the wrinkly outer layer of the brain that's responsible for all the high-level stuff, like language, abstract thought, and consciousness. In a rat, the cortex is about 30% of its brain. In us, it's a whopping 77%. This is the part of the brain that in many ways makes us human. Okay, so we've established we have this big, powerful, and very hungry organ. But what's it actually made of? Let's zoom right in and meet the 2 critical types of cells that make it all happen: the famous neurons and their absolutely indispensable support crew, the glial cells. And here it is, the star of the show, the neuron. This is the main signaling unit of the entire nervous system. And just look at its unique shape. It's not just some random blob, right? Its structure is specifically divided into these different zones, and each one has a very specific job in receiving, processing, and sending information. Information flows through a neuron like it's on a strict one-way street. It all starts at the dendrites, which act like these little antennas, collecting signals from other cells. All those signals then funnel into the cell body, which is kind of like the decision maker. If the total incoming message is strong enough, bam, a brand new electrical signal gets generated and sent speeding down the axon to talk to the next cell down the line. But here's something that's really crucial to understand. There's really no such thing as a typical neuron. Thanks to the groundbreaking work of Santiago Ramon y Cajal, you're looking at his incredible drawings here, we now know there are over 50 different types of neurons. They have all these wildly different shapes because they have different jobs, and they even speak differently. Some fire in these rhythmic bursts, while others fire in super rapid, continuous spikes. Now, neurons might get all the glory, but they absolutely cannot function alone. They depend on their amazing supporting cast, the glial cells. The name actually comes from the Greek word for glue, because scientists originally thought they were just simple filler, holding everything together. We now know their jobs are way more active and absolutely essential. There are three main types you should really know. First, you have microglia, which are basically the brain's dedicated immune system, fighting off invaders. Then there are oligodendrocytes. Think of them as the brain's electricians. They produce the myelin insulation that wraps around axons, which is critical for signal speed. And finally, you have astrocytes. These are the multitasking caretakers, doing everything from providing nutrients to helping with signaling to even forming scar tissue after an injury. Okay, now we get to the electricity. This is where it gets really cool. But before a neuron can fire off a signal, it has to be, charged and ready. It needs a stable electrical baseline. The best analogy is a drawn bowstring, just holding all that potential energy. In neuroscience, this is called the resting membrane potential. So what in the world is a membrane potential? Well, it's just a fancy word for voltage. Seriously, just like a battery has a positive and a negative terminal, a neuron has an electrical difference between the inside of the cell and the outside. And this voltage is created by very carefully separating charged particles, which you call ions. And this separation is super specific. The key players are positive potassium ions, which are kept at a really high concentration inside the cell, and positive sodium ions, which are packed on the outside of the cell. Now, critically, the inside of the cell also has all these large, negatively charged proteins and phosphates that are just too big to get out. This imbalance of charge is the entire foundation of the neuron's electrical potential. The net result of this precise ion setup is absolutely crucial. Because of those trapped negative proteins inside, the neuron ends up with a stable negative charge compared to the outside. This resting voltage sits at about minus 70 millivolts. And that negative potential, that's the stored energy. That is our drawn bowstring, just waiting for the signal to fire. Okay, our baseline is set, the bowstring is drawn, we are ready for the main event. Let's talk about the action potential, the fundamental electrical zap that neurons use to communicate. This, right here, is the language of the entire nervous system. So how does a neuron actually release that stored energy? Well, the secret lies in these amazing specialized proteins called ion channels. The best way to think of them is like tiny, highly specific gates that are embedded in the cell's membrane. When these gates swing open, they let certain ions flood across, and it's that sudden rush of charge that instantly changes the membrane's voltage. But what's the key that unlocks these gates? Well, there are three main triggers. Voltage-gated channels, which are key for sending a signal down the line, pop open when the voltage around them changes. Ligand-gated channels open when a chemical, like a neurotransmitter from another neuron, binds to them. And then you have stretch-gated channels, which respond to physical force, like the touch receptors in your skin. Now, to talk about these voltage changes like a pro, we need a few specific terms. When the inside of the cell becomes less negative, moving up towards zero, we call that depolarization. When it starts heading back down to its negative resting state, that's repolarization. And if it overshoots the resting state and becomes even more negative for a moment, we call that hyperpolarization. This brings us to the formal definition of an action potential. It is an incredibly fast, we're talking just 1 or 2 milliseconds, and complete reversal of the membrane voltage. But here's the key. It is not a graded response. It's a threshold event. A neuron will only fire if the initial depolarization is strong enough to cross a critical tipping point, which is usually around minus 50 millivolts. If you cross that line, an action potential is guaranteed to happen. And the scale of this reversal is just immense. In that 1 millisecond, the voltage doesn't just go to zero. It skyrockets from its resting state of minus 70 millivolts all the way up to a positive 30 millivolts. That's a total swing of 100 millivolts before it plummets back down. This leads us to one of the most important concepts in all of neuroscience, the all-or-none principle. An action potential is not like a dimmer switch. It is a standard on-off light switch. It either fires at its full characteristic strength or it doesn't fire at all. There's no in-between. And what's more, that signal's size doesn't get weaker as it travels down the axon. This is what ensures your brain gets high-fidelity information even over long distances. An all-or-none signal is perfect for reliable communication. But the brain also has this relentless need for speed. So how did the nervous system evolve to make sure these signals travel as fast as humanly or, well, biologically possible? It turns out there are two main strategies for sending a signal. In simpler, unmyelinated axons, the signal has to be regenerated at every single point along the axon. It works,
but it's pretty slow because you have to constantly stop and reboost the signal. The more advanced solution is myelination, where those glial cells wrap the axon in a fatty insulation. Think of it like the plastic coating on a wire. This insulation stops the electrical signal from leaking out, and all the ion channels are clustered at these little gaps called the nodes of Ranvier. This clever structure allows for a process called saltatory conduction. Instead of having to regenerate at every single point, the electrical current just flows passively and super quickly under the insulated myelin, and the action potential only has to be regenerated at the nodes. So in essence, the signal jumps from one node to the next. It's a brilliant biological shortcut.
And the benefit of this jumping is a massive increase in speed. A myelinated signal can travel up to 100 meters per second. That's over 260 miles per hour. A signal can get from your brain to your big toe in a tiny fraction of a second. The exact speed gets fine-tuned by two things. A thicker myelin sheath and a wider axon both mean a faster signal. Wow. We've journeyed from the scale of the entire brain all the way down to the movement of single ions. So let's just take a moment to wrap up by crystallizing everything we've talked about into the core principles of how a neuron actually works.
All right, here are the absolute key takeaways. First, a neuron shape directly reflects its job, with specific zones for input, integration, and output. Second, all neurons maintain a resting electrical gradient, just like a charged battery waiting to be used. Third, this gradient is created by the very careful separation of different ions across the membrane. And finally, the speed of that signal gets supercharged by two physical factors, myelination and the diameter of the axon.
And that brings us to our final thought. We've just spent all this time learning that the fundamental unit of information in the brain is a simple, identical, all-or-none electrical spike. So the profound question, the one that keeps neuroscientists up at night, is this. How do billions of these identical little zaps, firing in incredibly complex patterns, combine to create the richness of human thought, the persistence of memory, and the deep well of our emotions? That right there is the magic and the mystery of the brain.